Hi everybody, it's a real pleasure to publish this new video for you. But before it starts, I wanted to give you the context of what you will see. So this video is a part of the eDNA Protist workshop that we organized a few months ago. In this video, we will cover different topics related to high-throughput sequencing, such tag jumping, spacer, purification, co-amplification, or the question of ASV versus OTUs. The discussion will be animated by my colleague Guillaume Lentendu from the University of Neuchâtel in Switzerland. This discussion will be quite long, so there is a chapter in the description of the video that will allow you to jump directly of the parts that you are interested in. I hope that you will enjoy and that you will learn some stuff on how to treat the mysterious eDNA.
If anyone have a specific question on this library preparation, PCRs and sequencing stuff, then we will take the different step one by one. But yeah, for the tag jump, actually I can share the screen. I have one slide on that. Yeah, tag jump is really common. Actually depends on how you prepare your library, but if you use single tag combination, like for one sample you use only F1 R1, second F2 R2, and third F3 R3, when you pull all your PCR product after the purification and you build a library with them, you will have tag jump because you have some denaturation steps and a rebinding of the double-strand DNA into some kind of chimeric sequences, and they will actually exchange their tag. So all combination of tag could be generated. And even so, sequence which have F2 R2 at the beginning could end up with F3 R3. So it's a really reduced amount of reads which will do that, but it could represent like 1 to 5% of the total number of reads of one Illumina mice run, for example. And yeah, in this paper, they show they use only the unique combinations. So here in green, it's reverse tag, and then on the x-axis, on the y-axis is the forward tag. And they use only the unique combination, so F1 with R1, F2 with R2. So you see in dark blue that's the combination they use. But almost all other combinations, they have been found in the raw sequences. So it's fairly easy to remove this light blue combination because they are not expected to. So no samples in your study have this barcode. But you can already see that if you use all possible combination of the barcode, so in place of using here the 20 to 25 unique combination, you just order 5 barcode and you use the 5 times 5, so the 25 combination of this 5 forward and 5 reverse, then you will have a much more amount of tag jump. So that's something really to take care of. So one solution is to use unique pair of tags. If you have more than 25 samples you want to pull in one read, then you have to do multiple libraries. It costs a bit more, like 30, 50 euro, dollar per library, but it avoids a lot of this tag jump. Another solution is to add positive control. That's something we are currently testing. So if you have a sample like mock community or a positive control of few species, then what we observed is that the total number of the reads of these positive ASVs, which come from species not expected to be in the sample you're interested in. For example, if you work with sole, then your positive control would be marine species. So that if you find any occurrence of these ASVs in any of your samples, then you know it's originates from tag jump. And here the strong correlation I've observed is between the total number of reads of each of these positive ASVs or OTUs and the maximum read count it was in soil sample in that case, so which are due to tag jump. So more an ASV is abundant, more it have chance to produce tag jumps. And if you have a positive control, then you can create this linear model and set up a threshold which relate to the total number of reads of one ASV. And if you know that, for example, here, if you have 100,000 reads for an ASV, then every occurrence in one sample which have less than 50 reads, it will be only due to tag jump. So you can remove all these occurrences, and it reduces a lot this false discovery of ASVs.
I'm sorry, what was positive control in this case?
It was 2 marine algae, microalgae, and we figured out there was also contamination by amib in these cultures. So it was 3 species which end up creating 12 ASVs, and they are purely marine species. And the samples analyzed in that case were soil samples. So we know that if ASVs assigned to this species were found in the soil sample, they could only come from the tag jump from the positive colonies. My few advice would be to have at least 2 to 5 nucleotide long spacers in front of the tags, so on the MIDs. So you have the spacers, which have different sizes, the tag, and then the primer on both sides, so with both the forward and the reverse primers. Then the idea of the spacers that it just adds heterogeneity of the position of the primer. So when the forward primers start to be read by the Illumina machine, because if all sequence have A at the 10th position, then you have a smear effect and the quality of your sequence will drop. So if the A of the first base of the forward primer starts at cycle 10 and cycle 11 and cycle 12 and cycle 14 and 13, you will reduce this smear effect. So only 1/4 or 1/3 of your sequence will have this this first base of the forward primers at the same time.
And you could have the same spacer for different tags. That's not a problem.
And actually, you can even, so what we are doing is just adding ends. So you have 2N, 3N, 4N. When you order primers with N, actually, at the company which produce the primers, actually N is, they add T, C, and G, and you will have the same amount of A, T, C, and G at that position for all the primer in the mix. And that add first some heterogeneity in these first positions, but it will also, depending whether you have 2, 3, 4 nucleotide in your spacer, and in the pool of your different samples with 2, 3, and 4 heterogeneity spacers will allow to have this starting position of the forward or reverse primer to not be at the same cycle. And that will increase the average quality of all your sequencing.
And should you control that it's not creating some dimer or some artifacts?
No, because you just ask for N. You cannot control for that. So if you have 4N in front of your tag, then you have all possible combination of 4 times 4 nucleotides.
Okay.
We'll create all combination. Of course, it could anneal badly with your forward primer or reverse primer and do dimer, but on 4 nucleotides, there's no chance that it's impact your the success of your PCR.
Okay, excellent.
But sometimes you will have tags which will not work well. Can come back again on the spacers. If you want to be as precise as possible, the idea is to maximize the base entropy, I mean the diversity of base at each position, and you can actually compute that in R, or so that to be sure that you have always 25% of each base at each position. That's also a way to select for your barcodes. So that's actually the spacers this heterogeneity spacer when you order the primers actually ends. So the company changed them with ATCG, so you will have as much ATC and G at that position for this primer. Yeah, the game is to choose your barcodes from a set of barcodes which have at least 5 nucleotide differences, and to optimize the entropy. That means how much A, how much C, how much G and T you have per position. So at position 3, at position 4, and so on and so forth. For example, here I've computed that. The display not so well, but for each position here from 1 to 12, you can compute how many A, how many T, how many C, how many G you have, and the entropy. So entropy value of 2 is the maximum. It's Shannon entropy, actually. And it's maximum when you have even distribution of the 4 bases. And when you start to be less even, then you reduce the entropy. And actually you can, so what I've done is to design loop and select the barcodes and the length of the spacer attached to each barcode, to shuffle them many times until you reach the highest average entropy. That's a bit going a lot in the detail, but you don't necessarily need to do that to add your heterogeneity spacer, but that's actually the background idea, is to maximize the entropy at each position to allow for better quality of sequencing.
Yeah, I just have a comment on that, on this tag or barcode design that you mentioned.
Yeah.
Usually the way we do here is to also take into account the template specific sequence that are in 5' end of your primer.
Yeah.
So we try to avoid, let's say, if there is on the first position in 5' after or before the beginning of the priming site, if there is an A, for instance, we don't put an A in a barcode.
Yeah, sure.
To be like as much as possible anti-complementary with a template, and to avoid having any kind of amplification bias.
Yeah, but that's also the way. I haven't shown it, but I had, when I have the spacer of 2, then I had the 2 first base of the primer at the end of the barcode, just to look at that. Yeah. Also at the last position.
Yeah.
That's right. I'm doing that too. Just to go really.
Yeah, for the nerds.
Exactly.
In the lab, we work with CO1, and with CO1 we have some problem, and we had heterogeneity spacers. So like your priming spacer specific to CO1, and after one sequence with N, another primer with N, another primer with 1 to 4 N.
Yeah.
So in fact, you command 4 primer for one tag, and you do this with all the reverse primer and all the forward primer.
Okay, but
so it's a lot.
Actually, it's not necessary to have for one primer to have 4.
Yes, it depends.
Actually, you can only have the first primer pair. So let's say you have a F1R1 tag, and you say for F1R1, then you have 2 spacers, and then for F2R2 you have 3 spacers, 3 nucleotide spacers, and so on and so forth. So you don't need to have all the 4 lengths of spacer for the same primer, for the same pair of tags, because then you need to order 4 times the same pair of tag, and I don't know, I see that just as a waste of money. I don't think it will improve the quality, because at the end it's just when all the sequences are loaded on the Illumina machine, they don't care from which sample they are coming from. So
yeah, because then when you order this target primer with spacer, what do? You do separate PCR for each couple of primer, or you mix all the primer before?
Yes, we made a mix for one tag,
But as Guillaume say, I'm not sure you will improve your signal by adding several different primer with different spacer.
They do this method, then try this, and it's working because they had a lot of problem with CO1. Now it's okay because there was not sufficient variability. Variability. I'm sorry for my English. So now we do this not for 16S, but okay, so CO1 we do this.
I would like to ask, could you please explain a little bit about purification options?
Purification after the PCR.
Yes, after the PCR.
Yeah, so the different option is either you purify on a gel, then you have a gel purification kit. You do your first PCR to control that your PCR amplification works. Most of the time you do replicate PCR. For gel purification, we were pooling and then loading 15 microliter, I think, on a big gel. You cut just the slice of gel where the size of the fragment you want to amplify is located, so where you have the band, and then you purify based on this bit of gel you just cut. So there's a kit where you can just put the gel on it. It's also a filter base. It allows really to be extremely pure. You don't need to go that complicated. So other alternatives are the magnetic bits base purification. So some will do one purification per sample. That's the standard protocols. Other way is to already do your pool. So you pool, you do your replicates, but also all the samples you want to have in one library, and then you do the purification. Like that, you reduce the costs. Because any other sequence from the different sample, they will be pooled together to do the library. So you can pool before or after. Of course, the earlier you pool, the most chance you can have to create tight jumps, for example. But this will be only created if you increase the temperature. So if the double strand DNA start to separate into single strand DNA. And the last option we have is actually colon filtration. That's one I personally use. So you have purification kit for DNA, which are just based on a membrane. It captures the DNA. It's just like, so the same process as for the DNA extraction. It captures the DNA in the membrane, you wash it with some product, and then you
And then what would be the best option in case the PCR products were not very rich? So I don't get that much DNA. And I guess during purification, I will lose partially some of that, isn't it?
I'm not sure you are losing much of the DNA during the purification. Actually, when you work with this colon, so if you pool before, so if you have, for example, 20 samples in one library, and you do your equimolar pool before the purification, then you have larger volumes that you purify, and then you have less chance to lose when they are pulled together. When you do the purification per sample, which is actually also a bit better if you want to have exact equimolar pools, because when you purify, you remove all the primers, dimers, which can impact the quantification of the DNA. But yeah, as soon as you load all your sample on the colon filter, you won't lose much. Because there were actually the first way to create libraries were to, at the start of Illumina, or the way it's proposed by companies, often is one library per sample. So you are doing one PCR with already the sequencing primer attached. It's indexed sequencing primers. So that means the Illumina machine will already do the demultiplexing by itself. And you get one library, that means one set of sequence per sample from the machine as raw reads. And that's also a way to avoid tag jump, but it's much more expensive. So the way to pool samples into one library is the way to go to be more.
It means if we, for example, send our samples for sequences, it will be in any case mixed from many different other samples, and this mistake will be more or less avoided.
If you have not enough samples to fill out a full sequencing run on a machine, they will use samples from other projects, but they create separate libraries. That means the machine will definitely separate the raw reads. So all samples from your project with sample of other projects. So that should not interfere at all, and you will not have tag jumps from other projects. I have one question in the list still from Magali Schweitzer.
Yeah.
And she was asking whether it's preferable to use PCR-free library to avoid the amplification biases.
So yes, but PCR-free library, as I understand, it's at the end when you do your library preparation.
Yeah.
But you still do the PCR for your targeted barcode. So you have the second PCR, not the first one.
Yeah, I am talking about the second one. Actually, it's when you have your first amplification, when you add a second amplification of 12 cycle, is it really good or is it best to avoid?
For me, if you can avoid another cycle of PCR amplification, it's really great, especially for us with forum, as it's a small groups. The sequence are really similar, and maybe Guillaume speak about this smear effect about spacer things that we add to avoid that. So if you put more cycle, I'm sure it can blur your system again. So I prefer, but depend in which company you want to sequence also. Because what we did with Yann, it's PCR free, but with in Angers, they did the second PCR, right?
Yeah, I was asking because I did both. With Yann, I was doing the PCR free, and then in Angers, we have the 12 cycles because the indexing is done in the second PCR too.
Yeah.
Can you do the indexing per sample?
We do the indexing with the second PCR.
Okay, so each sample have a different index.
Yeah, but just in the second PCR. With the other method, like they do in Geneva, you index in the first PCR, the only PCR, in fact.
Okay.
Yeah, it's just now in Angers, we have two different ways, the two different kind of data sets. So we have two different DNA extraction kits, because we were in the middle of this COVID things and they didn't produce the Chiagen thing. So we should find another one. So we have another DNA extraction kit, and we sequence in another company. So we should think and compare maybe the result we get a bit more deeper for these questions.
So typically, I won't say it will. I can't imagine it have a big impact. So both protocols have been tested, and they provide solid results. So these two approach, what David say, it's easier to use one PCR. The drawback is that with longer primers, the annealing success could be reduced because you have really long primers when you attach the sequencing primers in the first PCR, and the success of the amplification could be reduced. So particularly for groups which already have lots of mismatches in the primer region, it could have an impact. So there's actually three ways to prepare the library. The last way is to do the PCR with the sequencing primers and using a ligation kit, which are originally for metagenomic. So they were using the poly-A tail of metagenomic multiple displacement amplification to do the ligation of the adapters, so of the primer, of the sequencing primer after the preparation of the amplification and cutting of the DNA from metagenomic sample or metatranscriptomic. And that's where you can pull more samples, you can create libraries for multiple samples, you can reduce the costs. And that's the paper David had in his presentation. So they are showing the protocol for meta-barcoding with the ligation. It's the TAG-STD protocol. So you have like that the best success possible with PCR without sequencing primers. You do your 35 or 40 cycles with only the primers, the biological primers with the tags, and then you do the library preparation with a ligation step actually, which ligates the forward and the reverse sequencing primer after the PCR. But you still have so when you do that, that means you pull together multiple samples in one library, and you will have this issue of tag jumping that you need to take care of. So either with adding positive control or with mock communities, so to have at least samples in which you can test for how much tag jump you get in your sequencing.
I guess I had a question about primers. I know that it probably depends on your sample and what you're targeting and things like that, and how you're sequencing, but is the field leaning in any particular way for the best universal primer for protists? So I am just like, I'm coming from the 16S world, so I'm doing my first protist PCRs in the new weeks, and so we're testing a V4 and a V9 primer on our samples. But I'm just wondering, you know, like for 16S with the Earth Microbiome Project, they kind of do have some standard primers that a lot of people are using, and I know the PR2 website, but and there's hundreds of primers on there, and so I'm just wondering, are we coming to a consensus, or is everyone just using different primers, whatever works for our samples? Or
actually, so the PR2, it's the PR2 primer website. Actually, PR2 primer is published, so the paper describing the website, but there's another primer where they look at the different primers, and they are doing comparison and selecting the most, advising on the best primers to use. So far, actually, it's primers targeting the V4 region or the V9 regions, but they have all set of primers. They will have some amplification preferences for different groups. And typically, what you can do is to dig deep into the different groups you could be interested, or which are probably the most abundant in your sample, and control that it doesn't miss most of your diversity.
But it's also depend on your samples. It was tested that for V9 was a perfect one, but in our lab for our samples, it's V4 was the best one. I think it should be some research done before, at least for best primer for you, that you think that they are best. Maybe that's the way how to deal with this.
Yeah, to do in silico analysis definitely needed, so that you know which one is working with you. And actually, so here was for a conference on tested amoeba, so I've used the most commonly used primer for V4 region. So it's one of the two regions for all Protist meta-barcoding. And you can dig down to some taxonomic groups to know how well they are covered. Here's the genus level on the left side, and you see that for some genera, you have more than 5 mismatches on the forward or reverse primer region, so they won't be amplified. Other groups, they will have really long fragments, so they will probably not be sequenced, or well, so they will not end up in your clean sequence at the end of your bioinformatic pipeline, because you will not be able to do the pair end join, typical for Illumina sequencing. you have to know actually what you want to, or what you are expecting from your sample, and do this in silico analysis to get some expectation of what you will be able to cover and what not. Actually, for the foraminifers, I think they're quite badly covered by this V4 primers, and that's why there was lots of development of specific primers.
So if you don't know what's in your sample, then you need to use multiple primers, sequence them, and then determine which ones you should sequence.
Actually, you have so many you have so many data sets already available online, so that you can also try to look at previous paper published on the same habitat you are looking for, and get an idea on what kind of taxonomic diversity is covered by such primers.
Maybe I can just add something on that, because I have soil samples and I was trying to use the V4 regions at some point to amplify it, and I did some tests with the PCR. So I tested for the V9 and the V4. The V9 was perfect. I was so happy to see my small PCR fragments coming out, and it was V4, V5 region, and that didn't work at all. And I tried different stuff, different primers, and I almost got crazy in the lab. So it may work as in silico, but then when you go to the lab, you may actually get into the problem that at some point for me was like, okay, I don't have time anymore to waste on that. So I'll just go with the V9 and hope it works. So I guess that also may happen depending on your samples, so that you have to test them, so test in the PCR to see if you actually do get a fragment.
Using specific primer during my PhD thesis, I was using different set of specific primers, and also developed for Secozone, for the 454 pure sequencing, and for the ciliates. Actually, the common, so now the way to go with ciliates is to do nested PCRs. So you go with ciliate specific primers first, and then you amplify only the V4 inside the longer fragment amplified by the specific primer, which is, I think, the case also with primers for Secozone. And what happened with the ciliates, actually for the 454, we didn't need to do the nested, because it could sequence much longer fragments. But I think the forward primer specific to the ciliate, they create an homopolymer, an exact homopolymer. That means the forward primer is binding to itself quite strongly. But with 454, you had to attach at the 3' and already the sequencing primers. And the primers, they just elongate against themselves, and they were immobilized, and no PCR amplified at all. And when you realize that, and you have ordered all your primers costs like 500 euros, then it's a bit disappointing. So that's why it's really important to test your primers also in silico and with the adapters and the barcodes before ordering them also, because you will get the list of barcodes in many publications. Earth Microbiome Project, I think they have standard protocols with standard barcodes, but could be that some barcodes won't work with your primers because they are creating heterodimers or homodimers with your primers.
Yeah, now we're here speaking about library preparation, and I was considering doing it myself with my samples, but at the end we just send the DNA directly to the company. And I was wondering, how is it like a terrible idea to do, or if it's good because then everything is standardized, or what are really the benefits of doing everything yourself, or it just depends a bit of your samples? And if
so the experience I have experience with both way to send samples. Actually, depends the company if they have a dedicated workflow for meta barcoding. Most company, they will only propose you the standard 16S and ITS that is certified by Illumina in a way, and that's it. And some company, they propose more in-depth services. I won't make advertisements, but there's one in Switzerland we are working with quite a lot, and they have dedicated workflow for meta barcoding. And actually, when you work with samples, you get good quality DNA, and the organisms you target are easy to amplify. Then sometimes it's easier, so it's quicker, easier to directly send the DNA extract to the company. You want to go for all bacteria, all fungi, all protists, and do some biodiversity study, just send the DNA extract to the company and they will do all for you. Particularly if you have a lot of samples, like it's a PhD project, and the PhD is alone in the lab to do all the, so if you have, it's always depend also the scale of your lab. So if you have lots of multi-channel pipettes and then lots of thermocyclers and you can do process lots in parallel, that's fine. If you have lots of samples, but you're only with a single pipette and you have to fill 3 to 4 96 well plates, then it takes ages. And in terms of price, reduce the price when you have a lot of samples. So when you buy the biggest DNA extraction kit, when you buy the biggest PCR master mixes and so on, for the purification too. On the other hand, if you work with more targets which are more difficult to amplify, if you develop new primers, you definitely have to do it all by yourself. So that means the PCRs and the purifications. Again, here you have multiple ways to do purification. 10 years ago, we were doing purification on gel, cutting the gel on the good size to be sure we don't have the primer dimers and some wrong amplification in the product. Now we typically pull the triplicates together and do some column-based purification of PCR products. You have also the alternative of doing the purification with the magnetic beads, which could be a bit more expensive. There's still lots of paths to optimize your workflow, and depends how well your lab is equipped.
Yeah, so it's really a bit of a trade-off and also depending. So if I'm interested in general in the protist diversity, then I could easily go for just sending the DNA. But if I'm really focused in one specific group, then I better invest more time, I guess, to get the right stuff.
The experience we have too is that when you are not sure you will amplify your target group, redoing PCR it will cost a lot by companies. They will charge every new try, and if you want to have at least triplicates or duplicates for each samples, and it will cost a lot to optimize with a company. Well, if you need to do optimization, it's better to do on low scale, on few samples first, and then.
Okay, thanks.
If you go for a company, they will always ask you how much sequence you want per sample. So the way to choose that is to have an idea how much reads will help you to saturate the diversity of your sample. There was papers for bacteria which were estimating that already many years ago, and I think for beta diversity they were saying already it was for PacBio sequencing, so the first generation high throughput sequencer. And they were claiming that 1,000 reads is enough to have a good idea of the beta diversity. And if you want to saturate alpha diversity, you have to go for 10,000 reads. With Protist, my experience is that 10,000 reads is a bit low. So you should rather aim for 100,000 reads, raw reads, considering that you lose half of the reads by the bioinformatic processing, depending on the quality of the sequencing. We pull until 120 samples per Illumina MySec runs. So you get 15 million good reads, and it ends up with an average of 50 to 100,000 of reads per sample. So, and that's enough to saturate. Any experience so far with Nanopore. What I know is that it's produced reads which are much longer than the Illumina. So of course it has many advantages to resolve the species. I think it's the way forward too. We have the tendency to be happy with the methods which is well established, even if provide us not the full information we would expect. That means to resolve the community composition at the species level. that's not what's possible so far with Illumina. The reads, they are too short to resolve at species level. So Oxford Nanopore or PacBio, they could sequence, for example, the full ribosomal DNA operon, so from 18S to 28S for the eukaryotes. And with that, you have more than enough variation in the nucleotide composition to resolve the species level. So there's definitely advantage in terms of scientific question. In terms of cost, it's still not the same throughputness than Illumina. So if you go on a high-sec, if you're fragments, if you interest in Protist and you go for the V9 18S, you can go for high-sec and you get 100 million of reads and you have more throughputness but less resolution. So there's kind of balance to choose between the two approaches, depending the question you would have.
I've heard some things about Nanopore, just that it's really sensitive to strings of the same nucleotide. So if you have like AAAs,
homopolymers,
yes, then it's prone to errors.
Yeah.
And so I think with like 16S, you can get some data looking at the sequencing quality of Illumina versus Nanopore, because I know I'm looking at for longer reads too. And so I was thinking about Nanopore, but my advisor is kind of like, I don't know if I would go with that. You get longer reads, you know, but you get a lot more errors.
So yeah, there was this issue when they started with this third generation sequencing techniques. So there's still the issue of the quality. Of course, Illumina still have the best quality for one read. The alternative we are currently planning in our lab is to use PacBio, because with PacBio you can, so it circularizes the sequences and it can do multiple runs of sequencing on the same DNA molecule. On the first run, so if you do only one run of sequencing with PacBio, you have 30% error, or it was 5%, perhaps. So if you increase the number of circular sequencing, then you can drop the error rate to the same amount as Illumina. So if you go for PacBio with three rounds of sequencing, you're on the same error rate than Illumina. I think that's the only third generation which allowed to decrease that much the error rate. And of course, every technique will have its own set of bioinformatic approach to correct this kind of errors. Tomorrow, David will show you the pipeline with Data2 to create ASVs. So it's an error model-based correction of reads, which was optimized for Illumina. They also say it works well for PacBio. Of course, for Nanopore, if you say the homopolymers is the most numerous amount of errors, then perhaps you can take advantage of software developed for the 454 PRF sequencing, which was the first generation of high throughput sequencing machine, and they had the same kind of errors. So elongation or reduction of the homopolymers, and they have lots of softwares developed to correct these kind of errors. Or that's probably already available for Nanopore datasets. I haven't made the literature review myself for such tool, but I'm sure you can correct for the quality of this. And of course, yeah, you have this co-amplification. So the second part of the question of Heike is about the co-amplification. At least with the V4 primers, you have lots of co-amplification of animal, plants, and fungi. That could be a big issue or not. So you will see tomorrow we have typically a search dataset with 90% of co-amplification of plants. That's not optimal, but you can still work with it. But so keep in mind that if you are working with samples associated with one particular organisms, you can always design blocking oligos or DNA clamps, which prevent the amplification of the host DNA. So for example, people analyzing gut content, they will use such technique to block the amplification of the hosts, of the metazoa. For feces content, you can also block the co-amplification of the host DNA because it's only one species, so it's relatively easy to design these clamps, which are actually primers, so they should overlap the primer annealing region, and they have a special end which prevents the amplification once this clamp anneal to the host DNA.
Could it be a solution? Because we were thinking to amplify, the protist of the rhizosphere, but also the roots. And the problem with the roots, if we use universal primers, we will have mostly plants.
So that's typically a solution. Yeah.
And how did you call this? I don't know.
It's PNA clamps. You find it also under the name blocking oligos. There's no.
Also based on some mismatch, you can just build some mismatch in your primer that will block two sequence roots. It just needs to be checked. There are many techniques to avoid it.
Yeah.
But you need to test with your object. If you want to have one plant or more, it also will be.
Yeah, most of the time what's happened is that you want to reuse the same primers already used for soil or for water. So some generic primers, you don't want to develop new primers. So that's the solution to avoid developing new primers which have more mismatches against the host you don't want to amplify. So going for this blocking oligos. There's no many literature for Protist on that topic. Perhaps there's one or two papers using that, but it's relatively common for DIT analysis. So gut content analysis of mammals when they want to look for parasites. There's multiple papers which have developed such approach to prevent the amplification of the host. Of course, it doesn't reduce to 0% the co-amplification, but sometimes from 50%. I have myself designed that. It was a study with colleagues in the University of Bremen in Germany, and they want to use fungal 18S primers because the ITS primers that don't cover the basal fungal groups, and they had a lot of co-amplification of protists, which is actually the opposite of what we are most interested. So I designed primers to block the amplification of protist groups like Stramenophiles, alveolates, and Rhizaria. It was kind of mitigate success because these groups are really diverse, and to block really diverse groups is really complicated because you don't have stretch of DNA exactly identical to all of these outgroup, so protist outgroups for the fungi, which will share the same DNA to design one blocking oligo. For some samples, so it was a marine fungi, we could reduce the co-amplification of Rhizaria by 50%. So the proportion of fungi moved from 20 to 70%, but not in all samples. It works, but depends on all. If you have only one plant species and you can design really perfect blocking oligos. If you have the rhizosphere of multiple plant species, then it will be a bit more tricky to get a good blocking oligo. So the quality is actually from the Illumina machine. It's when they read the sequence. So the sequence, they are on the Illumina, you have a plate on which all the DNA is loaded, and the DNA is actually in some small bubbles, and they will actually distribute all over the plates. And each of these single sequence will attach. So they have so-called sequencing primers, and these nucleotide strain, they will be used to attach the sequence to the plate. The plates have lots of single-strand DNA radicals, which allow a sequence to attach and to create a bridge. And then for each of attached sequence, they will do a PCR on the plate. So that means one sequence will be multiplied by 100 to 1,000 copies. in one specific location on the X and Y. During the sequencing reaction, they will add nucleotide one by one, and each nucleotide have a fluorescent dye attached to it. And the camera will scan each color after they add nucleotides. And that's like that they sequence by addition of color, which are detected always the same X and Y position. The quality is linked to how well the machine can translate the light signal into a nucleotide. Because what could happen is that two different sequences are near to each, or too near one to each other on this plate, and they have different light signals, and that will impact the quality of the reading. So if after the position 10, they have two different nucleotides, but they are located exactly at the same X and Y position, then you will have a blurred signal. So that could impact the quality. Another issue is which is specific to the meta-barcoding. It's the fact that all sequences have the same nucleotide from position 9 to 30, because they are all amplified with the same primers. And the issue is that when the machine come at the 10th nucleotides, it, for example, they give A, then it will be blue on all the plates, and you will have a so-called smear effect. And the machine could not discriminate between the sequence, because the intensity is like when you do a photo with overexposition. You have too much light, and you cannot discriminate the individual position of this many millions sequence or sequence cluster placed on the plate. And that will decrease the quality of the reads. And so how it translates exactly between the light intensity and the quality, I cannot tell you. That's software and the magic of the machine, I would say. But it's typically how they give a note on each position for each sequence to say it's the best quality we can have, or it's middle quality. So typically the qualities range from 0 to 40 or 41, depend the machine. 40 is the best, 0 is unknown. And yeah, during your bioinformatic processing, then you can filter based on the quality, based on the maximum expected error rate, for example. So more you accumulate low quality bases in a sequence, more you have a chance that one base is wrong. And you can use this information to filter bad quality sequences. But you will see that tomorrow. It's definitely an information we use to clean such datasets. I've used OTUs and ASV on the same datasets for 18S, and actually we've published a paper also, okay, 16S and 18S both. So on 18S ciliates, we show that you found the same result, that you have more ASVs than OTUs.
I had the same result as well. I also checked with the dataset ASV and OTUs, and I got more ASV because it's only one nucleotide difference, right?
Okay, yeah, sure. Yeah, it will depend the datasets. Yeah, of course, depends also how you create the OTUs. My experience was with using Swarm as clustering algorithm. Swarm is don't have a global threshold. Sure, when you use a 97%, 98% similarity threshold, you will group sequence from potentially different species together. Swarm is a clustering algorithms which is based on network, more or less network. It binds all sequence with one nucleotide, 1 to 2 nucleotide sequence differences. It will also separate sequence link like that with 1 to 2 nucleotide differences if there's two highly abundant sequences with like 10 nucleotide difference, but some sequence linking them, they will split it to two OTUs based on the abundance. So it avoids the issue of grouping sequence which potentially originate from different species.
So what's the difference with ASV or ESV?
So ASV, it's not only based on nucleotide, so the number of different nucleotides, it's also building an error model on, it's also using the abundance of, so the most abundant reads will be taken as a ground truth, and it will look at all of sequence around that most abundant true sequences and try to create an error model of how often A translate to C, how often A to G, A to T, and so on. And it will apply this error model to all the sequence then and create these ASVs. I haven't go totally in deep in the algorithms of the ASV, but yeah, my personal impression is that it's doing kind of clustering. So it's still creating OTUs, so operational taxonomic units, but at the variant level.
Would you say that SWAM is more appropriate for Protist than, because ASV was developed first for bacteria, 16S, but how does it translate to Protist, knowing all we know about Protist?
The experience is that you have this barcoding gap, which is very variable between clades and even between genus in the same family. And that's a big issue. So we were fine with using global thresholds 10 years ago because it was the best available solution, but it still lumped together a sequence from different species, even some different genera. On the other side, it will separate sometimes variants from the same species. At least SWARM is able to avoid the lumping together of sequence from different species into one OTU. So that's why my experience is that SWARM is producing more OTUs than ASV, but it was based on mock communities. So the increase in number of OTUs was mainly due to artifact OTUs not being grouped back together with the original sequence. So Illumina sequence will produce not much, but it will produce point mutation, so artifacts with point mutation, and then it can create multiple lower abundance variants of one original sequence with 1, 2, 3, 4 nucleotides different. Actually, both approaches, SWARM and ASV, are comparable, but ASVs will SWARM clustering will still keep some low abundant OTUs, which are most likely artifacts, and then you will need to use some sort of thresholds to remove them from the dataset.
When we compare the 97% similarity and 100% similarity for SB, so for my knowledge, I say SB should be lower than the OTU number because of the similarity percentage. But why the SB are more? I don't know actually.
Yeah, because ASV is at the variant level, so you are not lumping together species which have really high similarities. When you use 18S, the 18S, so the V4, the V9 fragment, it doesn't have enough nucleotides to discriminate species. Not, at least not all clades. So some clades, they will have, so inside the genus, all species will have the same V4 region or the V9 region, or they will have only 1, 2 nucleotide difference, 100 nucleotide long fragments. So if you use the V9, 99% similarity clustering, then you will lump together sequence from different genera together, or different species from the same genera. The same at the V4, so the fragment is 400 nucleotide long. 97% similarity, it means 12 nucleotide differences. So you allow until 12 nucleotide difference for the same OTU. So that means you potentially bring a lot of species together. That you can actually look in silico, do some in silico analysis, and look for the similarity, the average similarity in different clades. For the fragment you amplified, and you will have an idea of what is the barcoding gap. But it varies a lot. I have done similar analyses, for example, for ITS and for 18S, and sometimes from family to family in the same clade, then the species, the barcoding gap, it's at 99%, and sometimes it's 96%. So it's really this 97% similarity threshold is really a big average for all bacteria. For the V4 Protist, I tend to use 98% similarity as threshold when I was using threshold, but it's still a big average. So that's the issue of global threshold clustering. Then when you go to the ASVs, you avoid that. And so you are keeping really separated between species. And often could be that you have also different copies of the same gene in one species with slight variations. So you can also end up looking at intraspecific variation. David, Enrique, some advice to contribute your libraries?
I really think you are the specialist here, so I'll let you deal with these questions. It's totally fine. It's you that talk first to me about this kind of issues. So yeah, so thank you for that, and I'm sure we can improve the overall quality by taking these kind of things into account in the future. That doesn't mean that our previous data sets are useless. It just means that our future data set will be better. That's it. Yeah.
We can expect, actually, as the tag jump seems to be quite common, except when you have the sequencing primer in the first PCRs.
Yeah, you can expect that you have this tag jumping, so you should be a bit careful when you interpret your data.
It will actually blur a bit your patterns. That means
it blur your patterns, and the main conclusion, right? We have everything everywhere, but maybe it's not very true because you have this tag jumping.
Yeah.
So from an ecological point of view, if you pay attention about all this kind of stuff we discussed here, you will just have a much better signal in your model. So it will just improve your results. That's it. But still, we find something with a data set that were not well designed, but that was well designed at the time we did that. So, but there is still some patterns that we can observe, so it's still okay. But if we redo this experiment now with the new methods, I'm sure we will find even better results. So yeah.
Yeah, great. But actually the right way to go before was to just set an abundance threshold. So if you don't have this positive control and you fear that you have tag jumps, or you already found samples assigned to tags which you didn't pull in the mix. So like F1 R2, there's no sample with F1 R2, but you still have 0.5% of your reads in your run which are assigned to this pair of tag, then you know you have tag jump.
Yeah.
And if you don't have samples to control for how much that represents in your other samples, then the best is to set a abundance threshold, so to remove the low abundant OTUs or ASVs.
That's the things we can do, and that's my advice when you treat your data set is, when you sequence, you always has this long tail of diversity, and to avoid the misinterpretation, I recommend to work with the most abundant ASV or OTUs, and don't consider too much these tails, especially with all data sets, because you have this tag jumping issues, PCR bias, sequencing error, and all these kind of things occur more often in these states. That doesn't mean that it doesn't occur in abundant ASV, but less. So it's why I recommend to fix a threshold to remove these states. And then with the new methods, you can consider the tails because you avoid all these kind of errors. And also the clustering algorithms now are really much better to deal with the sequencing errors. And yeah, so you reduce all this effect, and that's the kind of improvements we can assume we will have in the future. So yeah, I'm looking forward for that too. Okay, so if there is no more question, I think we can close the workshop for today. What we will do now is we will send it to you a link to download the file for tomorrow. And yeah, let's see tomorrow at 9 o'clock from Paris time. For those who want to dig more in bioinformatics, assignation, and statistics. So I would like really to thank Enrique and Guillaume for this very nice workshop. And thank you for your attention. It was really nice to have so many people interested by these topics. That was quite amazing. And I'm looking forward to see you tomorrow for the next part of this workshop. So I let the word to Guillaume and Enrique if you want to add something.
I was going to say just thank you to everybody. We are quite surprised to see how such a crowd here, but okay, and we hope that we met your expectations, and let's see tomorrow we'll be more focused on more technical issues.
Yeah, have a good sleep. Be ready for art coding day.